Assalamu alaikum, everyone. Today, inshallah, we're going to continue learning about the replicative cycles of viruses. So viruses, they can increase their number, or they can replicate or reproduce only inside cells. Okay? This is why they are considered obligate intracellular parasites. And each virus can infect certain types of cells. And this is why each virus has a host range. So the host range is basically represent the type and of the cells that can be infected by a virus. Okay? Some viruses, they infect humans, and not all, and even the viruses that infect the humans, some viruses that can infect only certain types of cells. Some viruses infect respiratory cells, but they cannot infect intestinal cells, for example. Some viruses, they can infect nerve cells. Some viruses, they can infect immune system cells. For example, just like the HIV virus or the human immunodeficiency virus. The human immunodeficiency virus causing AIDS can infect immune system cells, for example, only. Some viruses, they can infect plants. Some viruses, they can infect other types of animals. And some viruses even can infect bacteria. So every virus has a replicative cycle. Okay? And the replicative cycle begins by inserting the viral genome into the host cell. Now, insertion of the viral genome happens by different means or by different ways. But once the viral genome is inside the host cell, the virus will hijack the biosynthesis machinery inside the cell. So the virus will take the enzymes found in the cell, the ribosomes, the tRNAs, amino acids, and the virus will start building or expressing its own genes to build its own viral proteins. Also, viral genomes will be replicated, sometimes using the enzymes inside the cell, sometimes using enzymes encoded by the viral genes. So the viral infection begins. So this is a cartoon showing a general and simplified viral replicative cycle. دورة حياة التكاثر. So here, infection begins when the virus binds to the surface of the cell. And typically, on the surface of the cell, there will be some receptors that could be recognized by, let's say, so this is a receptor on the host cell, okay? And this receptor could be recognized and bound by proteins, maybe, on the surface of the virus, okay? So after the virus enters, for example, this is a virus, and here we have the genetic materials inside the virus, and the capsid is surrounding the gene. After entry of the virus genome or genetic material is released from the capsid, and now the viral, for example, DNA, in this case, will be replicated, and the genes of the virus will be expressed to produce capsid proteins, for example. And then the next step is the assembly between the viral proteins and the viral genomes or viral replicated DNA to produce new virus particles or new virus progeny. And these viruses can exit the cell in different ways, and they can now infect other cells. Of course, different viruses, they have different genetic material, they have different structures, they have different ways of entering the cell, and different mechanisms are used to replicate the genomes of viruses. And there are different viruses, they have different ways of being released from the cell. Okay, so this is a generalized and simple replicative cycle. So the first replicative cycle we're going to discuss in details is the replicative cycle of phages. Phages, they are one of the best understood of all viruses because a lot of research have been done on phages. Actually, much of, or let's say, huge deal of information of molecular mechanisms have been first discovered in phages and bacteria. So whatever we know currently about, for example, translation, transcription, DNA replication in eukaryotes or in humans, actually the information about these important mechanisms in humans and other organisms was obtained by studies, by what we've learned from the way phages infect bacteria and how bacteria carry out translation, transcription, and DNA replication. So scientists, they discovered or uncovered key molecular mechanisms by doing experiments on phages and bacteria. Okay? And then they compared or applied what they have learned about phages and bacteria to other animals, including humans. So phages, they have two alternative reproductive cycles, the lytic cycle and the lysogenic cycle. So the lytic cycle begins by the attachment of the virus on the surface of the cell. Okay. Here, the tail fibers are used for the binding of the virus on the bacteria. Of course, bacteriophages, they are much smaller than they are depicted here. They're just, this is not the right scale of the virus size to the size of the bacteria. A DNA virus, for example, will inject its genetic material, and the host chromosomal DNA will be degraded. The next step is that the replication of the viral genome and expression of the genes from the virus to produce viral proteins. And then we have self-assembly. Self-assembly means the different parts, the different proteins of the phage, they are assembled with the genome of the phage. So now we have a process called phage assembly. And then the new phage particles or phage progeny will be released from the cell, causing the lysis of the cell, okay, or disruption of the cell wall and the death of the cell. Okay, this is the lytic cycle. It's called lytic cycle, lytic from lysis, because it will end up with killing the bacterial host or killing or lysing the cells. Okay. In the lysogenic cycle, from its name, so here lyso means lysis. Genic, the genetic material of the virus will be at some point incorporated into the host chromosome. Okay. So viruses or phages that reproduce only by the lytic cycle, they are called virulent phages. Okay. In the lysogenic cycle, we have the virus injects its DNA, and what happens is that the phage DNA will circularize, and then it will integrate into the bacterial chromosome. So each time the cell divides, it must replicate its chromosome. At the same time, it will replicate the phage DNA, which is now called prophage. So the phage DNA incorporated or integrated into the bacterial chromosome is called prophage. Each time the cell divides, it will first replicate its chromosomal DNA and the prophage DNA. Okay. In some situations, or at some point, this is the lysogenic cycle. It's called lysogenic because here still the virus did not cause the death of the lysosomes. But at some point, the prophage might exit the bacterial chromosome and the lytic cycle will be initiated or induced, culminating with the production of phage progeny and the release of phage progeny after cell lysis. That's why it's called lysogenic cycle. It begins by integrating the virus genome into the bacterial chromosomal DNA, but at some points, or occasionally, sometimes, the prophage might exit the chromosome and the lytic cycle will be induced. Okay. Viruses that uses both the lytic and the lysogenic cycles are called temperate phages. Now, what causes a phage to go from the lysogenic into the lytic cycle is some environmental signals. Okay. So sometimes, for example, if you treat the cells with UV, the prophage will sense that there, of course, UV causes damage to the DNA. So the prophage will sense that the DNA of the cell is being damaged. So it will try to escape, and it will, the UV, for example, can induce or initiate the entry into the lytic cycle. This is, for example, an environmental signal, إشارات B. Okay. So some environmental signals like exposure of bacteria to UV light or to any other harmful chemicals might trigger the exit of the prophage and the entrance into the lytic cycle. Of course, bacteria, just like humans, so we humans have an immune system that protect us against viruses or invading viruses. Also, bacteria, they have their own mechanisms that will help them protect or will help them basically prevent viral infections. So we have the bacteria, they have defense mechanisms against viruses. And one of the defense mechanisms relies on restriction enzymes. So remember, restriction enzymes, these are enzymes that can cut DNA. And usually these resection enzymes, which nowadays we use them for cloning, they have been discovered in bacteria. Naturally, resection enzymes, they represent a bacterial defense mechanism against phages. So bacteria uses resection enzymes to cut the phage DNA. So if, for example, if this phage, like here, if the phage injects its DNA and there are resection enzymes, these resection enzymes will attack the phage DNA, the injected restriction. These resection enzymes will attack and degrade the phage DNA. If the phage DNA is degraded, the bacteria will be safe and it will not be affected by the virus. Okay. But these resection enzymes, why do they how they can specifically attack the phage DNA, but they cannot attack the chromosomal DNA? Why? Why do you think? Because these resection enzymes, they cannot differentiate between the phage DNA and the chromosomal DNA. If there is on any, so the phage DNA, if the phage DNA contains a restriction site for this enzyme, okay, it will be cut by the restriction enzyme. If the chromosomal DNA also contains a restriction site for this restriction enzyme, it will be also cut by the restriction enzyme. But actually, the restriction enzymes, they do not attack or cut the bacterial DNA. Why? Why do you think? Do you know why? Can you guess why? حذروا. So here in this case, the phage, the chromosome, the bacterial chromosome is not cut. Because the bacterial chromosome or the bacterial DNA is methylated. Okay, and if the resection sites within the bacterial chromosomes are methylated, this methylation protects the DNA from being cut by the resection enzyme. But the phage DNA that enters the cell, if it's not methylated, it will be cut by the resection enzyme. Okay. So the bacterial DNA is protected from resection enzymes by being methylated. Another defense mechanism against phages is the CRISPR-Cas system. So the CRISPR-Cas system was first discovered in bacteria, just like resection enzymes. And it turned out that bacteria use the CRISPR-Cas system to protect themselves against viruses. CRISPR, this acronym stands for clustered, regularly interspaced, short palindromic repeats. Okay. So in CRISPR, the bacterial genome, okay, so this is the bacterial, let's say, genome. The bacterial genome contains a region called the CRISPR region, or bacterial chromosome, or DNA, or genome, it's the same thing. Contains a CRISPR region. Now, the CRISPR region consists of two of repeats that are shown here. And so this is, here we have repeated DNA. Okay, usually between 20 to 30 nucleotides. Okay. And these repeats are called spacers. Okay. Sorry, it's not spacers. No, these are repeats. Between these repeats, we have spacers. So these repeats are actually DNA chromosomal, bacterial chromosomal DNA. And between these repeats, we have the spacers. And these are actually DNA from phages. Okay? DNA from phages or from previous infection by phages. So remember here, for example, when the resection enzymes, they cut the virus DNA, some DNA from some fragments from the virus genome are integrated into the chromosomal, into the bacterial chromosome inside the CRISPR region. So this is the DNA from a virus, okay, that previously tried to infect this cell. This is another DNA from another phage that previously tried to infect us. So you can think of a green phage, for example, injected its DNA, but its DNA was cut into fragments, maybe by resection enzymes or by any other. And now some of that DNA was added to the CRISPR region. Here we have a green, yellow DNA from another phage. So they could come from different phages. Okay. Between them, we have the repeats. So here we have a cluster تجمع, cluster of interspersed. So let me go back here. So clustered, regularly interspersed short palindromic repeats. So CRISPR stands for clustered, regularly interspaced short palindromic repeats. I just want to explain to you why it's called like this, because this name explains the structure of this region. Clustered, regularly clustered, we have a cluster of repeats and spacers. So these are called, these phage sequences are called spacers between the repeats. And we have the repeats. These repeats, which are given here, the same color, indicates that they have the same sequence. So clustered, regularly interspaced short palindromic repeats. These repeats are short palindromic repeats. They are short because they are made of maybe 20 to 30 approximately nucleotides or base pairs. Palindromic because just like resection sites, you can read the sequence, okay, from the top strand from 5' to 3', and from the bottom strand the same way. Interspaced, regularly interspaced, there are spacers between these repeats on a regular basis. So we have repeat, spacer, repeat, spacer. So between repeats, we have regular spacers. But these spacers are actually phage DNA. They're not bacterial DNA. That's why it's called clusterly regularly spaced short penetrable fields. Now, this region can be transcribed to produce one long RNA transcript. Okay? And this long RNA transcript could be processed by some enzymes, Cas enzymes, to produce what we call guide RNA. Guide RNA molecule. Each guide RNA contains an RNA transcribed from a repeat and an RNA from the phage. So this RNA transcript is cut into smaller transcript like this. So there will be a transcript with another repeat and a green phage DNA. Another transcript, a repeat, okay, and maybe here. And the yellow phage RNA. Okay. All of these are called guide RNAs. Why they are called guide? Because this guide RNA can bind to a Cas protein called Cas9 mainly. And now we have Cas protein RNA complex. Now this RNA is complementary to a DNA from the invading phage. Let's say that this phage, this is the DNA from this phage, and some sequences on this DNA are complementary to this DNA that is already in the bacterial chromosome. So this means that this RNA, the blue RNA sequence, and typically here, this blue sequence is around 20 nucleotides in length. Okay, so this is called guide RNA. It is called guide. Guide means dalil. The dalil siyahi, because this guide RNA will allow the Cas protein to bind to this DNA, target DNA region. Okay, so this is the DNA from the invading region, and this region of the DNA is called the target sequence that is complementary. So this is the virus target sequence that will be cut by the Cas9. So Cas9, with the help of the guide RNA, will bind to this sequence of the phage. Why? Because the RNA sequence is complementary to a sequence on the phage DNA. Once the binding happens, this Cas9 protein is an enzyme called nuclease. Nuclease means can cut nucleic acids. So it will cut the phage DNA, and if the phage DNA is cut, eventually it will be degraded, and the phage DNA can no longer replicate it or transcribed or expressed. Okay, which means the phage infection will not happen. Okay? These are so using CRISPR-Cas system, the bacteria can protect itself from invading phages by destroying the phage DNA, just like the resection enzymes do. All right. So now let's talk about the replicative cycles of animal viruses. Let's take 5 minutes break before that, and we will continue, inshallah, after at 12:10. So next, we're going to see some example replicative cycles of animal viruses, viruses that can infect animals, including humans. So viruses that infect animals are classified based on their nucleic acid. So the nucleic acid could be, or the genome of the animal virus could be RNA or DNA, and could be single-stranded RNA or double-stranded RNA, or single-stranded or double-stranded single-stranded DNA or double-stranded. Also, the viruses, some animal viruses have a membrane envelope, some do not. Okay. Typically, bacteriophages, they do not have an envelope or an RNA genome. So most of bacteriophages, they don't have an envelope or they do not have an RNA genome, but some they do. Okay. Unlike bacteriophages, many viruses that infect animals, including humans, they have membranous envelope. Okay. And this envelope contains glycoproteins. So proteins plus carbohydrates. Glycoproteins means, remember what you've learned in carbohydrates, or they have sugars or monosaccharides attached to these proteins. Now, this envelope surrounding viruses, usually it is taken or derived from the host cell plasma membrane. Okay. Let me go back here. As the virus, for some viruses, as they leave the cell, they will take some of the membrane from the host cell. So if these viruses might be surrounded by envelopes, and these envelopes actually, they are taken from the host. So here we have host membrane. But these envelopes, they might contain some of the virus glycoproteins, okay? Or viral proteins. So these are viral glycoproteins, okay? So these viral glycoproteins, they are not found normally on the surface of the host cell. So this is a replicative cycle of an enveloped RNA virus. Let me just, before we discuss some example replicative cycle, I want to discuss with you this table. So this table shows the classes of animal viruses. There are 4 major classes: double-stranded DNA, viruses with double-stranded DNA, viruses with single-stranded DNA, virus with double-stranded RNA, viruses with single-stranded RNA, and there are 2 types of single-stranded RNA. Ones with an RNA that will be basically served as an mRNA and will be used directly for protein production, or it will be translated directly. Some single-stranded RNA viruses, their mRNA cannot be used directly. It must be replicated first, okay? And it must be first used as a template for mRNA synthesis, and the newly synthesized mRNA will be translated. So here we have this mRNA is called, this single-stranded RNA is called positive sense. Sense RNA. And this one is called negative sense RNA. And sometimes they are given this symbol. So positive single-stranded RNA. And here we have negative sense single-stranded RNA. Okay. And we have group number 6 consists of retroviruses or single-stranded RNA that serves as a template for DNA synthesis. Okay. So some double-stranded DNA viruses, they have envelopes, some they do not. Examples of these viruses are the pox virus that cause smallpox, jadari. We have single-stranded viruses. For example, we have the parvovirus that cause some types of rashes, and boop. Double-stranded RNA viruses, we have the reoviruses like the rotavirus that, and this virus does not have an envelope. These are just example viruses. Rotavirus, this virus is very active during the summer and causes diarrhea in kids. هذا بسبب اسهالات في الأطفال في الصيف. Single-stranded positive sense RNA viruses like the coronavirus. So the coronavirus and is considered an envelope virus. It contains a single-stranded RNA genome, and it is a positive sense single-stranded RNA, which means it could be used directly for protein production. It could be directly retranslated. Example of negative sense single-stranded RNA viruses are the Ebola virus. This is a very dangerous virus. It's a deadly virus that have been that caused some problems in West African countries. Okay? The influenza virus, for example, is a negative sense single-stranded RNA virus. Rabies virus and measles also. Hospa is an example of negative sense RNA virus. Retroviruses, like the human immunodeficiency virus that causes AIDS, is an example of a virus that is enveloped, and its RNA must be used as a template for DNA. Okay. So now we're going to look at some example replicative cycles of animal viruses. So here we have the replicative cycle of an enveloped RNA virus. So the RNA is surrounded by a capsid, and the capsid is surrounded by an envelope, and the envelope contains these glycoproteins that will bind to certain receptors. Okay, so this is a glycoprotein receptor. Okay. Tala, what's the difference between positive and negative? Here, the RNA of the virus, it's called positive if it is used directly as an mRNA or used as an mRNA and could be translated directly. Negative, this means that the RNA of the virus cannot be used as an mRNA, cannot be translated directly. It will be used as a template to synthesize a complementary copy of the viral RNA genome, and that copy is used as an mRNA for synthesis of proteins. This is the difference between negative and positive. So here we have the viral RNA. The viral RNA genome is not used as an mRNA. It is used as a template for the synthesis of another RNA molecule, and this RNA molecule will serve as an mRNA and it will be translated. So here we have a negative sense single-stranded RNA. At the same time, the RNA genome will be replicated. The mRNA synthesized from the viral genome will be translated to produce capsid proteins in the cytoplasm and to produce glycoproteins inside the endoplasmic reticulum. And then these glycoproteins will be transported by transport vehicles into Golgi, and from Golgi, transport vehicles will transport the glycoprotein into the membrane, the outer membrane of the cell. Now, inside the cytoplasm, the capsid proteins will self-assembly with the replicated viral genome. And then as the viral capsid and the genome, or as the virus particle, the new virus particle, leaving the cell, it will take with it some of the membrane from the cell, and this membrane will contain the glycoproteins of the virus. So now we have a virus. Some viruses, they take membranes from the host nuclear envelope, not the plasma membrane. Okay? And then they replace them by an envelope made from the Golgi apparatus membrane. So some viruses, they take their envelope from the plasma membrane, the outer membrane of the cell, or from membranes inside organelles, or from the organelle membranes inside the cell. Retroviruses, they have single-stranded RNA, but also they have the enzyme, they use the enzyme reverse transcriptase to convert their RNA genome into DNA. An example of retroviruses, the human immune deficiency virus, or the HIV virus, that can cause AIDS, the acquired immune deficiency syndrome. So this is the replicative cycle of the HIV. The HIV consists of 2 RNA strands that are single-stranded and they are identical. And connected to each strand is an enzyme called reverse transcriptase. Now, these 2 RNA molecules are surrounded by capsid, and the capsid is surrounded by a viral envelope with the glycoproteins that will allow the virus to attach to the surface of the cell. Okay. Now, the HIV virus can infect immune system cells. Okay, so would be a human immune, let's say, immune system cell. Okay, so like we have HIV can infect a helper T cell, for example. Helper T cells, macrophages, خلايا البلعمة, and dendritic cells. خلايا التائية المساعدة and dendritic cells. Okay. And once the virus binds to the surface of the membrane, once the virus through its glycoprotein attached to a receptor on the surface of the cell, its envelope will fuse with the plasma membrane, releasing the capsid, and then inside the cell, the capsid will disintegrate, releasing the viral RNA. The viral RNA will be used as a template to make a DNA strand using the viral reverse transcriptase. This is very important. And then we have an RNA DNA hybrid. The DNA will be replicated. So now we have double-stranded DNA. And this double-stranded DNA enters the nucleus and it will be integrated into the cell chromosomal DNA. And now we have, just like prophages, we have provirus. Now this provirus can be transcribed, and the mRNA can be translated to produce viral capsid proteins. and reverse transcriptase, and glycoproteins in the ER, and the capsid proteins and the RNA genome also could be replicated, and they will assemble together to produce the capsid and the RNA. And as the virus is leaving the cell, it will take some of the cell membrane, and this cell membrane will contain also glycoproteins. So now the virus will be surrounded by an envelope. Okay. In the last concept, we're gonna discuss how viruses can cause disease. So viruses that can cause disease to humans, to animals, especially if we're talking about animals, farm animals that are important for us as a source of food, or even cause disease in plants, especially plants that are used as a source of food to humans and to other animals. So diseases caused by viral infections, they can affect humans, agricultural groups, محاصيل زراعية, and livestock worldwide. Livestock here, we're talking about حيوانات المزرعة. Okay. Now, we have also some infectious particles called prions, which are made only of proteins, can also cause disease in the plants and animals as well. So first, let's discuss viral disease in animals. Viruses, they can cause diseases because they damage cells, they kill cells, or sometimes by causing release of enzymes from the lysosome inside cells. Remember, the lysosome contains the lytic enzymes, the enzymes that can degrade proteins, nucleic acids, carbohydrates, and lipids. So if the lysosome is damaged by the virus, the enzymes inside the lysosome will start attacking every molecule inside the cell, causing the death of the cell. Okay? Some viruses, they infect cells and causes cells to produce toxins, and these toxins will lead to damage and lead to disease. Okay. So we typically have an immune system that protects us against viruses. But sometimes we have to train this immune system. So vaccines are used to train the immune system. Train means بندرب immune system. So vaccines are harmless derivatives, مشتقات, of the pathogenic microbes. So some parts of the virus that will be used to stimulate تحفيز or train. So train, stimulate, okay? Train and stimulate the immune system to protect us against the pathogen, okay? That's why we use vaccines to prevent certain types of viral ailments. So each one of you already took a number of vaccines during childhood. So you have been vaccinated against a number of viruses like poliovirus, شلل الأطفال, mumps, and many other types of viruses that can cause disease. Smallpox, for example, virus, okay? And many other types. So many many vaccines are given to kids uh, to protect them against many viral infections. So, for example, right now we are all taking the let's say the vaccine against the coronavirus. Now, these vaccines, some vaccines, they completely protect us against disease, but some vaccines, they cannot protect us against viral infections. They can just minimize the disease symptoms caused by the viral infections. So, for example, the coronavirus vaccines, they do not protect us against infection. You might be infected even if you are vaccinated, but it will protect you against going to the hospital. So people who took the vaccines, even if they are infected by the virus, they will not suffer from serious illness, or they do not have to go to the hospital. But if someone did not take the vaccine and he or she got the virus and they are infected, there is basically there's a high chance that they will have more serious illness or more serious symptoms, and they might have to go to the hospital. So vaccination against COVID-19 does not protect against infection. It will protect you from, or it will basically protect you from going to the hospital or from having serious symptoms or serious COVID-19 illness. Okay. So vaccines can prevent certain viral illness, but also some vaccines can minimize. They cannot prevent, they just minimize the severity of, so prevent or minimize the severity of viral illness. illness اللي هو المرض. illnesses. Okay. Some viral diseases could be treated by antiviral drugs. Okay, these antiviral drugs, they can, many of them, usually they will have the suffix ver at the end of the disease, at the end of the name of the virus, of the drug. So antiviral drugs usually they target the ability of the virus to replicate its DNA or to assemble its genome with other proteins. Okay. So again, these are not vaccines. We're not talking about vaccines. Vaccines are taken before having the viral infection. But these antiviral drugs are used after the viral infection happens. Okay. So, for example, there's a number of antiviral drugs that are currently being used, like remdesivir. Remdesivir is an antiviral drug again, these they do not cure. مش علاج شافي. They treat. They just are given to people with the viral disease to minimize the severity, قوة of the disease. Okay. So when you see a drug with VIR at the end, you have to understand that this is a drug used to treat a viral infection, okay, or a viral disease. Some viruses, they suddenly appear, just like the coronavirus. Coronavirus, nobody heard about it. Most people never heard about the coronavirus before 2019. But suddenly, this virus appeared, and now it's a pandemic. These are viruses that are called emerging viruses, which are normally found in nature, but they do not cause problems. But suddenly they might have mutations that will increase their ability to spread between organisms like humans. So, for example, the Ebola virus, the coronavirus, these are viruses that suddenly appeared. We have the Zika virus that appeared in 2015. Okay. And many other viruses. Okay. Also, the HIV virus is considered an emerging virus, and which suddenly appeared in 1980s. Okay. So some viruses are emerging viruses, some viruses are not emerging. So for example, the influenza virus is not an emerging virus because typically we are infected with this virus every winter. So a lot of people are infected with this virus during winters, and it doesn't cause serious problems, and the symptoms are self-limiting. Okay. Another example of emerging viruses are the basically the flu virus. I will the viruses that cause the swine flu or the bird flu. Okay. So in 2009, for example, a general outbreak which was endemic to Mexico and the United States. Epidemic, sorry. So epidemic, this means there's a lot of cases of diseases caused by the virus in a confined geographical area. So for example, in 2009, there was an epidemic of a flu-like illness in the United States and Mexico caused by the influenza virus named H1N1. Okay. So H1N1, it's an influenza virus A, and its common name is the swine flu. Okay, influenza الخنازير. So influenza viruses, there are let's say there are different types of influenza viruses. The type that causes, so there are basically three types. So we have influenza virus A, B, and C. Typically, A causes typically A causes, come on, causes infections in, let's say, in the wide range of animals like birds, pigs, khanazir, okay, horses, and sometimes animals, humans, sometimes humans. But B and C can infect humans only. So influenza B virus, influenza C virus, humans only. But sometimes an A virus infecting birds or pigs or other animals might also infect the humans, like the swine flu. It's called the swine flu because this is a virus, influenza A virus, that can infect pigs, but now it was transformed and it acquired the mutations, and now it can infect humans, or began to infect humans in 2001. Now, these influenza viruses, they are named based on the type of two proteins, okay? Heme agglutinin, and so H, this naming system, is based on two proteins that are found on the surface of the virus. So we have, so N or H stands for heme agglutinin. And N stands for neuroaminidase. Aminidase. Okay. So influenza viruses, they have, let's say, they have segmented double-stranded RNA. So if we go back to this table, the classification of the influenza viruses is that they are they have single-stranded. So they have where is the influenza virus? Yes. So they have single-stranded RNA, okay, and they have negative sense single-stranded RNA. Okay. In terms of the structure of the influenza virus, they have the capsid, okay, and inside there will be, I think if I'm not mistaken, there will be 8 segments of RNA. I think 8 segments of RNA molecule. Okay, so these are RNA segments. And on the surface, on the envelope of the virus, there will be 2 types of proteins. We already So we have, let's say, the neuraminidase and the heme agglutinin. Use this guy. So H and N. There are different types of H, they are given number, different types of N, they are also given. So this virus, the influenza virus, occasionally can be transformed to not only infect animals, but also to become and emerge as a virus that can infect humans as well. Okay. Now, if you remember, so in 1918, there was a very disastrous pandemic. Pandemic means a global epidemic. The virus is everywhere, in every country, in every continent, just like COVID-19. 1980, there was something called the Spanish flu, influenza الإسبانية. And this Spanish flu actually caused by H1N1, influenza A virus. Now, there are currently 16 different types of heme agglutinin. So 16 types of H. And there are 9 types of neuraminidase proteins discovered so far in influenza A viruses. Okay. So this heme agglutinin, for example, this is this protein helps the virus attach to the cell. To host cells, while this one helps the with the release of the virus from the cell. Okay, so both they are found in the surface of the influenza A virus, and each one has its own function. Did you hear also about the bird flu? Bird flu. This bird flu, I think, I don't know if you remember, it appeared first in 1997, okay, in Hong Kong, and it was caused by H5N1 influenza virus. Okay, so this bird flu or avian flu, usually found in birds, or this strain of the virus infects birds, but at some point, because of some mutations, it was transformed and it was able to infect birds and the humans at the same time. Now, how viruses can emerge, how viruses that are usually infecting animals become viruses that can infect humans and start spreading all over the world. Typically, these viruses are RNA viruses. And RNA viruses, they have high rate of mutations. And these mutations are the main cause of the emergence of these viruses. Okay. And also, other processes, another process that can contribute to the emergence of viruses is that these emerging viruses, typically they might be found in a small isolated human population, and then they could spread quickly around the world. Okay? Because now, you know, because of the technology and transportation, and you could basically within hours move from one continent into another, and because of you know the worldwide trade of merchandise and goods around the world, it is much easier nowadays for a virus to spread around the world in no time. This wasn't the case, let's say, 100 years ago, because 100 years ago, we did not have large ships or airplanes that could transport people between countries or between continents. Okay? And typically, 3/4 of a new human disease, they originate from animals. Okay. So this coronavirus, for example, it's a virus that is naturally found in bats and wild animals. At some point, this virus might have mutations that allowed it to infect humans. The HIV, for example, virus is a virus that can infect monkeys, and from monkeys, it was transferred to humans. We have the swine flu. It's an influenza A virus that can infect pigs. We have also the bird flu that used to, it's an influenza virus that used to, that can infect birds. And at some point, it started infecting humans. Okay. Of course, what causes the transformation of the virus? Sometimes mutations might change the virus. So now it can be transformed from a virus that infects animals and humans to a virus that can infect animals and humans at the same time. And scientists think that global climate change might have a consequence or might cause the spread of viruses through other organisms like mosquitoes. Okay. Like the Zika virus. The Zika virus was spread through mosquitoes, for example. خلال mosquitoes اللي هم الذباب والناموس. Okay. Of course, the global human activities, which allow the humans to be connected more easily through airplanes, trains, and ships. Also, every remote area that might have emerging viruses are now connected to cities by roads or railroads or by trains, by buses. So all these factors, they might contribute to the spread of viral diseases very quickly and on a global scale. So let me stop here. Inshallah, we will continue discussing viral diseases in plants and in animals tomorrow, inshallah.